Hi, hello. This is Ramesh Kumar Lalkota, Assistant Professor of Botany, MBS Government Arts and Science College Autonomous, Mehboob Nagar. In our last class, we saw about micro and macronutrients. Today, we will see enzymes in our class. So, what are enzymes? Enzymes are organic substances produced in the living cells. They are popularly known as biological catalysts, and they catalyze all the biochemical reactions in the living cells. In living cells, whatever be the biochemical reaction, wherever it may take place, but it requires a enzyme to catalyze it. What is the role of a enzyme? It brings down the activation energy and allows a number of substrate particles to turn into end product. Okay, so without these enzymes also the biochemical reactions takes place, but they'll take place at a very slow pace. You cannot imagine such reactions, and the life will be at a great uh, danger or unimaginable if we imagine that without catalyst, that is enzymes. Okay, so what do these enzymes do? Enzymes accelerate biochemical reactions without affecting its equilibrium. Biochemical reactions can occur in all living cells even without enzymes, as I said just now. Okay, so regarding its discovery, Louis Pasteur was the first person in 1857 who said that uh, yeast was a living organism, and fermentation was associated with life and growth of yeast. So Louis Pasteur was the first person to state that fermentation is associated with yeast cells. That is in 1857. And Traub in 1858, he said that fermentations are carried out by certain chemicals closely related to proteins produced only in living cells. That means fermentation is carried out by chemicals which are closely related to proteins. And in 1878, Kuhne was the person who coined the term enzyme for the ferments proposed earlier uh, by the investigators. Okay, and later on in 1887, Edward Buchner was the person who discovered that yeast extract could cause fermentation of grape juice, and he coined the term zymase for the active element involved in it. So, the first person to separate the enzyme from yeast cell is Edward Buchner. He discovered that fermentation causing yeast extract and named it as zymase. Later on in 1926, J.B. Sumner isolated the enzyme urease in crystalline form, for which he was awarded the Nobel Prize. That means urease was isolated by J.B. Sumner in its purest form, that is, in its crystalline form. Okay. So, where do these enzymes present? That is, distribution of enzymes. Where do these enzymes are synthesized? Enzymes are synthesized in the cells itself, but they may function inside the cell, that is intracellular, or they may function outside the cell, that is extracellular. Extracellular enzymes are also known as exoenzymes. Okay, so for example, in fungi and bacteria, for digesting the substances on which they grow, that means the fungi and most of the bacteria are saprophytes, so they have to digest the external food. They depend upon external food, organic matter. So for digestion of these food particles, these saprophytes synthesize some enzymes. So the action of these enzymes is extracellular. Hence, they are known as exoenzymes. In the same way, insectivorous plants like Drosera, Nepenthes, they also produce exoenzymes. And where do these enzymes present? Enzymes are present in specific sites within the cell. We all know that cells do have many organelles, and each organelle is associated with a specific function. For example, chloroplast is related to photosynthesis, mitochondria to respiration, Golgi apparatus for uh, as, fat lipid metabolism, endoplasmic reticulum for lipid metabolism, and lysosomes for lytic enzymes, glyoxysomes. So likewise, in the nucleus also some enzymes are present. Uh, uh, these nucleus contains enzymes of DNA and RNA metabolism. Cytoplasm contains enzymes involved in glycolysis, and this chloroplasts involve such as the enzymes meant for light reactions as well as dark reactions. Okay, so uh, within the cell, each and every organelle is associated with a specific function. So all these cell organelles, they produce the enzymes which are um, associated with their function. Okay. Then structure of the enzymes. What is the structure of the enzymes? Enzymes, we already said that they are proteins. They are nothing but they are proteins. They are formed in the same manner as the other proteins do. That is, how these proteins are formed and where they form. They are formed on ribosomes, on endoplasmic reticulum by the process called protein synthesis, which involves transcription and translation. Okay, so these enzymes are also proteins, but all proteins are not enzymes, but all enzymes are proteins. Okay, so enzymes are again of two types based on their structural composition. Simple enzymes and conjugated enzymes. What are simple enzymes? These are made up of only protein molecules. Only protein is seen in these enzymes. So only protein part is present. Uh, for example, trypsin, pepsin, amylase, all these are simple proteins with only protein part. Simple enzymes with only protein part. Then conjugated enzymes. They contain protein and non-protein part. Protein part is called apoenzyme and non-protein part is called prosthetic group. Protein part plus non-protein part together constitute holoenzyme. Okay, holoenzyme, apoenzyme and holoenzyme. Apoenzyme is only protein part, that is simple enzyme. If apoenzyme together with prosthetic group is nothing but non-protein part. Okay, together they constitute holoenzyme. So these holoenzymes based on the prosthetic group, they are again they show the prosthetic group may be inorganic or organic in conjugated enzymes, that is in holoenzymes. Uh, if the prosthetic group is inorganic, it is known as cofactor, and this attaches firmly and it cannot be separated through dialysis. It is so firmly attached that you cannot detach it from the apoenzyme. Okay, so for examples of cofactors are magnesium, manganese, ferric, ferrous, and zinc. These are some of the examples of different cofactors. Okay, then organic prosthetic groups are called coenzymes. They can be separated easily by dialysis, and the examples are NAD for dehydrogenases, NADP for dehydrogenases as well, and thymine pyrophosphate, that is TPP for pyruvic decarboxylase, and FAD for dehydrogenases. So these are some of the examples of coenzymes. Coenzymes are nothing but organic prosthetic groups, and cofactors are inorganic prosthetic groups. Examples for inorganic: magnesium, manganese, ferrous, zinc, and they cannot be separated through dialysis. Okay, where do you find this manganese? In enzymes such as phosphohydrolases, phosphotransferases. Okay, so this manganese is present as a cofactor or activator, metal activator. This cofactor is also known as metal activator. Zinc is seen in carbonic anhydrase, alcoholic dehydrogenase, carboxypeptidase, etc. And ferrous ferric ions are cofactors for catalase, cytochromes, peroxidases, ferridoxins, etc. And copper is a cofactor for tyrosinase, ascorbic acid oxidase, and potassium cytochrome oxidase, pyruvic kinase, molybdenum for nitrogenase. In such manner, a lot of metal activators or coenzymes act as cofactors act as a prosthetic group for conjugated enzymes. So, what are the different properties of these enzymes? Enzymes are chemically they are proteins and physically they are colloids. Chemically they are proteins means all enzymes are proteins. As I said earlier, all enzymes are proteins, but all proteins are not enzymes, and they are formed as the other proteins do, that is through transcription and translation. Transcription occurs in the nucleus, whereas the transcripted mRNA enters into cytoplasm where it undergoes translation producing proteins. The same way these enzymes are also formed in the same way. Physically they are colloids. Okay, they provide large surface area for the reactions. Uh, and the next one, soluble in water. They are soluble in water. Water enhances the activity of enzymes. Without water, enzymes do not participate in any biochemical reactions. Water enhances the activity of these enzymes, and enzymes on hydrolysis yield amino acids like proteins. They do form amino acids and hydrolysis. The next one is heat sensitive. That is, they are thermolabile. Uh, what is thermolabile or heat sensitive? At 0 degrees centigrade, they are inactive. The optimum temperature for their action is 25 to 30 degrees, and in human beings, it is 35 to 40 degrees. And above 45 degrees centigrade, they slowly start denature. Okay, at 60 degrees they completely denature, that is the structure gets destroyed and they move no more, they function as enzymes. Okay, then enzymes are sensitive to pH and temperature variations as well. They are inactivated by all factors which denature these proteins, that is strong acids, strong bases, they can deactivate these enzymes. And enzymes are capable of causing reversible reactions, that is single or two different enzymes may be involved in this reversibility. For example, glycerol reacting with fatty acid in the presence of lipase forms into fats, and in a reverse reaction these fats in the presence of lipase they turn into glycerol and fatty acids. In the same way, sucrose in the presence of a sucrase enzyme, it forms into glucose and fructose. And glucose plus fructose in the presence of phosphorylase enzyme converted into sucrose again. So these reactions are reversible. Okay, enzymes are capable of causing reversible reactions. Then required in very small concentrations. That is, um, they are required in extremely small concentrations to carry out the reaction. A single molecule of catalase can decompose 10 to the power of 6 atoms of H2O2 per minute. And sucrase catalyzes sucrose at the rate of 10 to the power of 5 molecules per minute. In the same way, alcohol dehydrogenase 10 to the power of 9 atoms per minute, and hexokinase 10 to the power of 11. And carbonic anhydrase, it has most the enzyme activity of this carbonic anhydrase is very uh, enormous, that is it can uh, catalyze six, uh, 6 lakhs of atoms of carbonic that is uh, the substrate molecules in a second. Carbonic anhydrase catalyzes 6 lakhs of substrate molecules per second. So this enzyme activity is represented by turnover number, that is TON, turnover number. What is turnover number? The number of molecules of substrate converted into end product in 1 minute by one molecule of enzyme is called turnover number. So different enzymes have different turnover number. Sucrase has 10 to the power of 5, catalase 10 to the power of 6, alcohol dehydrogenase 10 to the power of 9, hexokinase 10 to the power of 11, and most of all other enzymes, the most reactive or most catalytic action is seen in carbonic anhydrase, which can convert the substrate into end products at a rate of 6 lakh molecules per second. Okay. Then amphoteric in nature. What is amphoteric in nature? That means enzymes can react with both acids and alkaline substances. Okay. Whatever substance which reacts with both acids and alkalis are known as amphoteric substances. So these enzymes are also amphoteric in nature, and the protein part of these enzymes helps to react with cations as well as anions. Um, then enzyme activity can be inhibited by cyanides, sulfides, and etc. These cyanides and sulfides, in the presence of these substances, the enzyme activity can be inhibited. And these enzymes are substrate specific. Again, substrate specific means only maltase can act on maltose, sucrase can act on sucrose, and hexokinases, hexoses affecting phosphorylation has the enzyme hexokinase plus ATP. So these are group, that is, they are substrate specific. Only urease can act on urea, but it never acts on sucrose or cellulose or anything else. That is substrate specific. Again, they are group specific and stereo specific. Group specific means phosphorylations affect from hexoses affecting phosphorylation reactions. They have kinase plus ATP. Okay, so stereospecificity is nothing but enzymes show specificity to dextro and levo optical or structural isomers. Example, levo amino acid kinase. Certain enzymes may also show specificity to cis as well as trans isomers. For example, cis trans isomers of fumaric acid. So they are stereospecific and they are group specific. Stereospecific is nothing but dextro and levo optical or structural isomers. Example, levo amino acid kinase, and cis trans specific cis trans isomers. Example, cis trans isomers of fumaric acid. And enzymes closely resemble inorganic catalysts, but they accelerate or speed up the rate of reaction, mostly in positive manner. That is, they never retard the rate of reaction. So this is about the properties of enzymes. The next topic, nomenclature and classification of enzymes. So enzymes are specific for particular reaction. So they are named after substrate on which it acts. For example, lipases acts on lipids, ureases acts on urea, maltases acts on maltose. Okay, so this is based on which it acts. And the next one is named after the reaction they catalyze, that is oxidation reactions are catalyzed by oxidases, reduction reactions are catalyzed by uh, reductases. Okay. And the popular classification of enzymes is done by the IUB, which was established in, which was given this classification. IUB, International Union of Biochemistry, a commission on enzymes, was organized in 1964, where they have given this uh, classification of the enzymes. Okay, so the commission established a numerical system of classification according to International Union of Biochemistry. Enzymes are divided into 6 classes, and each enzyme is given a code number with 4 digits. Okay, for example, the code number for an enzyme is 1.6.6.1. That is, four digits are seen in enzyme code. Okay, which is in which the first digit indicates class, second digit indicates a subclass, third digit indicates a sub-subclass, and the fourth digit indicates the serial number of the class. Okay, so as a whole, hmm, the enzyme code is represented by four digits. For example, 1.6.6.1. Okay, which shows that 1 is the class, 6 is the subclass in class 1, and 6 again is a subclass in subclass 6, and 1 is the serial number of that particular enzyme. Okay, so according to this IUB, these enzymes are classified into six groups. What are those six groups? The first one is oxidoreductases, second one transferases, third one lyases, hydrolases, then lyases, isomerases, and ligases are the different enzymes given by uh, the IUB classification. Okay, so what happens here? We'll see one by one. Oxidoreductases. The enzymes which catalyze oxidation and reduction reactions are included in this group. Okay, oxidase. What are oxidases? Oxidases are nothing but which add oxygen. Reductases which add hydrogen or electron. So, and dehydrogenases as well, which remove hydrogen. Okay, so oxidases, reductases, and example is dehydrogenase, which remove hydrogen or add oxygen. So they are included in oxidoreductases class 1. Then next one is class 2, transferases. The name itself indicates that they bring about the transfer of a group from one molecule to another molecule. Example, kinases, transaminases. Okay, glutamic acid plus oxaloacetic acid. Glutamic acid plus oxaloacetic acid in the presence of transaminase gives alpha ketoglutarate acid and aspartic acid. Initially, okay, so there is a transfer of amino group from glutamic acid to oxaloacetic acid, which ultimately gives rise to ketoglutaric acid, alpha ketoglutaric acid plus aspartic acid. Okay, so these are transferases. And one more example is hexokinase, which transfers ATP molecule from one molecule to other molecule. Then, next one is uh, hydrolases. What are these hydrolases? The third class, they catalyze hydrolysis reactions by adding water to a variety of bonds and generally help in the cleavage of the substrate. For example, any substrate plates, water gives rise to that substrate. What is that? Uh, with the hydrogen, that substrate with hydrogen, and OH molecules will be liberated from water. Water gets split into H plus and OH minus ions. H plus ions goes with the substrate and OH minus with the other part of the substrate. Okay, so example, carbohydrates, esterases, proteases, all these are hydrolases. Okay. Then the fourth one is lyases or lyases. Fourth one is lyases. These lyases, they remove the group from the substrate non-hydrolytically. That is, initially in hydrolysis, the substrate may split into two products, in which one has H plus ions and the other goes with the OH minus ions. But here, lyases, these enzymes help in the removal of groups from substrate non-hydrolytically. For example, fructose 1,6-bisphosphate in the presence of aldolase. Here it forms into two 3-carbon compounds, that is DHAP and phosphoglyceraldehyde, that is dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Okay, here no role of water, H plus or OH minus ions. So these lyases, they help in removal of groups from substrate without hydrolytically or non-hydrolytically. Okay. Uh, One more example is pyruvic acid. It splits into acetaldehyde plus carbon dioxide in the presence of pyruvic carboxylase enzyme. Okay, then the fifth class is isomerases. They catalyze isomeric reactions. For example, glucose 6 phosphate in the presence of hexose isomerase forms into fructose 6 phosphate. And the same way, levolactic acid in the presence of lactic racemase isomerase gives rise to dextrolactic acid. So, these isomerases help in isomeric reactions. And the last one, ligases. Ligases are these are also known as synthetases. These enzymes catalyze the synthesis of different types of bonds such as carbon-carbon bond, carbon-nitrogen bond, uh, and catalyze these enzymes catalyze the synthesis of uh, new formation of new bonds between carbon-carbon, carbon-nitrogen, carbon-sulfur, carbon-oxygen, etc. Okay, so synthesis of new bonds done in the presence of these ligases. For example, acetic acid in the presence of ATP and coenzyme forms into acetyl coenzyme A plus AMP and inorganic phosphate. Okay, so these are six classes of uh, enzymes as per the IUB classification: oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. Very important in view of your examination. Then the next part of our lesson is enzyme action. So what is enzyme action? So enzymes form an intimate union with the substrate upon which it acts. The surface of the enzyme contains active centers, active sites, which are very small areas on the surface where substrate molecules can join. You can see here, this is the enzyme, and here are substrate molecules. They have specific sites called active sites. Okay, so these active sites are very small. The region on the surface of the enzyme molecules which combines with the substrate is known as active site. There may be a single active site, or there may be several active sites on the same enzyme molecule. So two theories have been proposed here to explain the enzymatic action. One of them is lock and key model, and the other is induced fit theory. Okay, so the first one, lock and key model. This was proposed by Emil Fischer, and later on it was elaborated by Paul Fieldis and D.D. Wood. Okay. So, as per this theory, the enzyme and the substrate are spatially complementary to each other. They are complementary to each other. They fit together like a key fits in a lock. No other key can open a particular lock except its own. In the same way, these substrate particles, they fit in the active site as a key in a lock. So this theory is known as uh, lock and key theory, proposed by Emil Fischer. So an enzyme substrate complex is formed there, and the enzyme substrate complex reacts and forms the end products, and the enzyme itself is regenerated, and it won't undergo any change in its composition, in its structure, anything else. Whatever enzyme was there initially, the same enzyme will be regenerated at the end of the reaction. Okay, it reacts with the substrate molecule. It accelerates the rate of reaction. It turns the substrate molecules into uh, end products by forming substrate enzyme complexes, which ultimately form into end products. Okay, so the enzyme action is compared with the proper key fitting in a lock and opening it. This theory is supported by compounds similar to substrate. Okay, whatever different substances which are similar to the structure of this substrate molecule, they also go and fit into the active sites of a particular enzyme. Okay, these compounds compete with the substrate for active sites on the enzyme. They often inhibit the enzyme activity also. So, for instance, for example, the conversion of succinic acid into fumaric acid is catalyzed by the enzyme succinic dehydrogenase. The presence of malonic acid will inhibit the enzyme activity because the malonic acid has the same structure just as that of the succinic acid. So it goes and occupies the spaces on succinic dehydrogenase, uh, which doesn't have any active sites for this succinic acid. Okay, so these malonic acid, it goes and occupies all the active sites. So the rate of reaction will be very slow, or the reaction will be inhibited. Okay, so these are competitors with the same structure. The presence of malonic acid here, it will inhibit enzyme activity. Okay, so and again, if the concentration of the succinic acid is increased, then the enzyme activity will be restored. This phenomena is known as competitive inhibition. Okay. The next theory is induced fit model, and it was proposed by Coastland in 1958. Since the lock and key relationship does not explain all aspects of enzyme action, and because of the rigidity of the catalytic site in the model, this induced fit model has been proposed by Coastland. Okay. Uh, Earlier we saw that only a particular structure, or the substrate with a particular structure which is same as the top on the active site of enzyme, it can go and fit into the active site of the enzyme. But here the substrate molecule, it makes the the enzyme molecule makes some changes in its in its structure related to that of substrate structure, substrate molecule structure. Okay, so according to this principle, uh, the substrate induces a conformational change in the structure of the enzyme molecule. Okay, the substrate interacts with the reacting groups of the amino acids within the active sites, and resulting in the change of conformation of the enzyme. In this way, certain chemical groups of enzymes align on the enzyme in the correct spatial orientation for binding of the substrate. Okay, this phenomena is known as induced fit theory, because the active site is precisely complementary to the substrate only after the substrate is bound. So this is all for today. Tomorrow we will see enzyme kinematics. Okay, thank you very much.